In the last video, we gave a broad overview of sensation and perception. In this video, we'll talk more specifically about how sensation and perception work as they apply specifically to vision. First, we will cover the basic structure of the eye and discuss how light photons are transduced into neural signals. Then we will cover two key elements of visual perception: how we perceive color and how we perceive depth. The eye is the sensory organ that detects light, enabling us to see. At the front of your eye is a clear, dome-shaped covering called the cornea. The cornea helps protect your eye by keeping out germs and debris. The shape of the cornea also helps to filter out some ultraviolet rays. Beneath the cornea is the pupil, a small opening through which light photons can pass through. When light levels are low, such as at night, your pupil will dilate or expand to allow as much light as possible to enter. When light levels are high, such as in the middle of a sunny day, your pupil contracts. The pupil's size is controlled by muscles that are connected to the iris, which is the colored portion of your eye. Next, light passes through a clear, curved structure called the eye lens. When light passes through the lens, it bends or refracts. This actually helps to focus and direct the light to specialized cells in the retina at the back of your eye. Your eye lens can also automatically change its shape to focus on objects at different distances. However, in some individuals, the eye lens does not refract light properly, which can result in near or farsightedness. At the very back of your eye is your retina, which contains specialized nerve cells called photoreceptor cells that are activated by light photons. Specifically, photoreceptor cells fall into two different general categories: rod cells and cone cells. Rod and cone cells are so named because of their two different shapes. Cone cells are very sensitive to detail and are directly involved in our ability to perceive color, which we'll talk more about in a minute. Cone cells work best in bright light conditions, which is why it's hard to see the color of objects in dim light. Instead, everything just tends to look kind of gray. Rods, on the other hand, can't detect color and don't provide the same kind of detail as cone cells do. However, they are specialized to allow us to still see, even in relatively low light conditions. Overall, you have many more rod than cone cells in your eye. However, they are not distributed evenly throughout the retina. Rather, cone cells are generally more concentrated toward the center of your eye, and their highest concentration is in the fovea, which is a small depression in the retina. Because cone cells are more concentrated in the center of your eye, your visual acuity is highest when you are looking directly at something. In contrast, when we use the periphery of our vision, we are relying mostly on rod cells, so it's harder to detect both color and detail. You can try this out yourself if you want. Try holding up a colored marker on one side of your face, just so you can barely see it, and see if you can guess the color. You'll probably find this to be more challenging than you think. If the rod and cone cells are stimulated with enough light photons, they will fire an action potential, starting the process of transduction. This nerve impulse then travels through several layers of nerve cells, including horizontal cells, bipolar cells, and ganglion cells. The function of these additional cells is complex, but they appear to play a role in helping us to detect edges, contrast, and speed of motion. It's also likely that some level of color perception occurs at our ganglion cells, which we'll talk more about in a minute. Eventually, all of this visual information will be carried via these nerve impulses to the optic nerve. The optic nerve is actually a bundle of around 1 million nerve fibers, which are axons from the ganglion cells. At the optic chiasm, these nerve fibers actually cross over each other, so that information from your right visual field is sent to the left hemisphere of your brain, and vice versa. This follows the principle of contralateralization that we learned about in the previous module. After crossing the optic chiasm, visual information is sent via a number of brain structures, including the thalamus, eventually making its way to the occipital lobe for further processing. This begins the process of visual perception, that is, how we actually interpret and make sense of the visual input we receive from our eyes. In the rest of this video, I'll discuss two key aspects of visual perception: how we perceive color and how we perceive depth. First, however, let's pause for an application practice question. Imagine that Lucy is walking in a dimly lit street late at night. This means that Lucy is primarily relying on what to see? The answer here is A, rod cells. Remember, rod cells are what allow you to see in low light conditions. Earlier in the video, I mentioned that cone cells are what help us to perceive color. But how do they do this? This has been an area of intense research and even heated debate for many years. Today, most researchers agree on two leading theories that explain how we perceive color: trichromatic theory and opponent process theory. It turns out that neither theory is mutually exclusive. Instead, each theory appears to be correct in its own way, helping to explain specific phenomena related to color perception. Before we discuss each theory, however, we first need to remember that light within the visual spectrum is made up of different wavelengths. Light can have longer and shorter wavelengths than these, it's just that we can't detect them. Specifically, the color that we perceive as purple is made up of the shortest wavelengths that we can detect, around 400 nanometers, whereas the light that we perceive as red consists of the longest wavelengths that we can detect, around 700 nanometers. The trichromatic theory proposes that different cone cells are specialized to detect light at only certain wavelengths. Specifically, this theory proposes the existence of three different cone cells: one type of cone cell that only fires at shorter wavelengths, that is, in the blue range; another type of cone cell that only fires at middle wavelengths, that is, in the green range; and a third type that only fires at longer wavelengths, that is, in the red range. You might be wondering why only three types of cone cells. Is that enough to allow us to see the entire visual spectrum of color? Well, as it turns out, yes. As you can see here in this graph, red, green, and blue can be combined to make any color in the visual spectrum. For example, red and green can be combined to make yellow, red and blue can be combined to make purple, etc. The existence of three types of cone cells also helps to explain the condition of color blindness. Color blindness is a highly genetic condition that occurs in around 7 to 10% of men and less than 1% of women. Individuals who are colorblind have a reduced ability to see or distinguish between shades of color. Although some individuals cannot perceive color at all, this is actually pretty rare. Rather, more often, colorblind individuals have difficulty distinguishing between two hues. The most common type of color blindness is called red-green color blindness, where individuals have difficulty distinguishing between red and green. This can occur when a person's red or green cone cells either don't function properly or are missing entirely. Although the basic tenets of trichromatic theory are well supported, it turns out that this theory on its own doesn't fully explain the process of color vision. For that, we also need opponent process theory. Rather than taking place at the level of our cone cells, opponent process theory appears to happen in the ganglion cells that we discussed earlier. Specifically, opponent process theory proposes that color is coded via three opposing pairs of color receptors: red-green, blue-yellow, and black-white. Importantly, receptors can only send information about a single color at one point in time. So, for example, when the color red is being stimulated, then that means the color green is being suppressed, and vice versa. The same is true for the other two opposing pairs. Opponent process theory nicely explains a particular phenomenon called afterimages. Afterimages are an illusion where an image tends to linger in your vision even after the original stimulus is gone. Let's try this out. First, stare for about 30 to 60 seconds at this green and yellow flag, and then either close your eyes or stare immediately at a blank piece of paper. I'll give you a minute to pause the video and try this out if you'd like. So if you did this activity, what did you see? You should see an afterimage of the flag. However, instead of the flag being green and yellow like in the picture, the flag in the afterimage should appear to be red and blue. Why did this happen? Well, this is thought to be the result of opponent process. After staring at the flag for so long, your green receptors became tired from all this continuous firing, while the opponent color, in this case red, was continually being suppressed. The result is that when you close your eyes, the color that was being suppressed now becomes dominant, causing you to see what's called a negative afterimage, or an afterimage consisting of the opposite colors. Finally, let's talk about another important topic in visual perception: how we perceive depth. Depth perception is what allows you to see the world in 3D, understanding an object's size and distance from you. Although depth perception is obviously critical for navigating many everyday tasks, from pouring yourself a cup of coffee, to driving your car, to playing almost any kind of sport, it's actually pretty remarkable that our brains can do this. This is because the images that are captured on your retina are actually in 2D, similar to a photograph. Using top-down processing, your brain then has to reconstruct these images to create the 3D perception of your environment that you consciously experience. So how does your brain do this? Well, first, remember that you have not just one eye, but two. And because your left and right eye are in slightly different positions on your face, the images that they send will be ever so slightly different too. By effortlessly doing some complex math, your brain is able to compare the differences between these two images to help you perceive depth. This is called stereoscopic vision. And this is also the same principle behind 3D glasses. The glasses feed slightly different images to your left and right retinas, creating a 3D effect. Because stereoscopic vision uses both eyes, individuals who have had damage in one of their eyes often struggle with accurately perceiving depth. However, if you close one eye, your world doesn't suddenly become a flat 2D image. This is because your brain can also determine depth by relying on various contextual cues that it can receive from just a single retina, and these are called monocular depth cues. There are many different monocular depth cues that your brain relies on, and I'll highlight just a few of the main ones here. First, occlusion is the principle that an object in the foreground will occlude or block any objects behind it. So in the picture on the left, you might perceive the white triangle as being closer to you because it appears to be blocking a red rectangle behind it. Relative size is another monocular cue that your brain can use to gauge an object's distance from you. Your brain knows that images that are farther away from you will literally cast a smaller image on your retina. So for example, if your brain assumes that objects are all the same size, like the image on the left, then it will perceive the smaller objects in the right image as being farther away from you, rather than just literally being smaller. Finally, if you've ever taken an art class, you might have learned to draw two parallel lines like the image on the right, where they appear to converge or meet at a vanishing point on the horizon. This is called linear perspective, and it is another way that your brain can gauge depth. For example, in this image, each of these silhouettes are literally exactly the same size. That is, they are literally casting the same size onto your retina. However, using linear perspective, your brain assumes that the silhouette on the right must be farther away from you. So using the same relative size principle that we just discussed, your brain therefore makes an adjustment and assumes that this person must be really huge compared to the other two silhouettes in the picture. Because we rely on so many context cues to determine depth, this is also an area where our brains can be susceptible to some predictable errors. For example, maybe you've seen optical illusion street art before, where an artist paints a scene like this one that almost looks 3D, even though it's not. To accomplish this, the artist took advantage of our brain's well-trained monocular depth cues to trick us into perceiving a giant hole in the sidewalk when in fact there isn't one. For some additional examples of 3D street art, you can check out the supplemental video in Canvas. It turns out that your brain's use of depth cues is also probably the explanation behind the Mueller-Lyer illusion that we talked about in the very first video. Remember that although these two lines are in fact the exact same length, your brain perceives the one on the bottom to be longer. Why? Well, there's still some debate here, but one possible explanation is that in right-angled rooms, the angles in configuration typically corresponds to objects that are closer, whereas the angles out configuration typically corresponds to objects that are farther away. Therefore, using the same relative size principle that we just learned about, your brain automatically makes a correction for the size of the lines. Normally, this correction is perfectly appropriate, although in this particular case, it leads to an error. Let's pause for one last application practice question before we close this video. In the Mueller-Lyer illusion, the fact that we perceive one line as being shorter is primarily the result of... The answer is B, top-down processing. In this module, we covered the process of visual perception. For the exam, you should be able to identify the different structures in the eye and their function, describe the process of how energy from light photons is transduced into neural signals, describe the different functions of rod and cone cells, define and distinguish between trichromatic theory and opponent process theory, and explain how each theory contributes to our ability to perceive color, and discuss how your brain is able to perceive depth, including both stereoscopic vision and monocular depth cues.